Alright, welcome to our first video on transcription. And we're actually going to talk a little bit about DNA replication today as well. But what we want to think of is that we're talking about a process that we call, in biology, the central dogma. Which means that all living things have to follow this rule. And what that means is we're going to go from DNA, so we have our information in DNA. Right, that's the hereditary information. It's packed into the nucleus. It's what is passed from generation to generation. We're going to convert that to RNA, and then we'll take the message in the RNA and we will make protein. And so we call this the central dogma. And today we're just going to focus on this little bit here where we convert DNA to RNA, and that is transcription. Okay? So here's a cartoon of the process we're talking about, DNA being opened up. All right, each of these little lines is a base, right? So we have our complementary base pairing. We have to open it up, and we make complementary base pairing to make our RNA. And here's my RNA. We then take the RNA to a ribosome and we make protein. All right, so in your cells right now, you're making about 10,000 different proteins currently, which is pretty wild to think about. So what has to happen then is the cell turns on genes, right, and allows them to be opened up and transcribed, which then they become translated into proteins. Right? So those decisions have to be made so that you can function properly. So here we have basically a nucleoside we start out with. So here's a base, all right, here's my deoxyribose, and we do not have a phosphate group. Here's a nucleoside monophosphate, nucleoside diphosphate, nucleoside triphosphate. This one has the most energy. We will use the triphosphate forms when we synthesize RNA or DNA. We always use the triphosphate form. And that's because when we cleave this off, all right, we will, actually we're going to cleave it here, we cleave these off, we will release energy and allow the reaction to occur. So let's look at DNA first. We've talked a little bit about this. So here's the space filling model, but let's look at this on the right. What we see here is we have our major groove and our minor groove. And to transcribe or to replicate this, what has to happen is enzymes or proteins have to interact at both the major and minor grooves so that we can work with the DNA properly. So this is a little simpler diagram. So here what we have is we have one sugar phosphate backbone and another sugar phosphate backbone. They are anti-parallel, right? This one goes 5 to 3, this one goes 3 to 5. Then we have our bases, and they are hydrogen bonding in the middle, all right? And so what we're going to do is we're actually going to take one of these sides, let's say this one, and move it out in this direction, and then we will allow for the RNA to complementary base pair. Sounds pretty simple. If we were doing DNA replication, we peel both sides apart and we form what's called a replication fork, and we use complementary base pairing to make DNA. So let's look at that DNA process. We'll go into this in an awful lot of detail later in the course. But for now, what we want to know is that we use the parental strands that we're seeing here in dark blue. All right, those serve as a template, and to the template we make the complement. All right, and so here we're making a complement, and here we're making a complement. So that's the basic principle of DNA synthesis, and when we're done we will have a DNA molecule that is half new and half old. The DNA is too long for the cell, so it has to be packaged, and we do that by supercoiling or twisting up the strands. All right, so when we twist those up, you can see it takes up less space, right? It's not as long when we form what are called supercoils. Now, when we take the supercoiled DNA, we can also wrap it around proteins. All right, so here's a nucleosome core particle. All right, and we are going to basically wrap the DNA around these proteins and help it be packaged. All right, so let's look at transcription, right? That's really what today's focus is. So now what we have here is we are going to make RNA transcript, we always make RNA or DNA in the 5' to 3' direction. So the new strand will always be like so. Or if we were going the other direction, all right, if we were using the non-template strand down here, we would go this direction. All right. So here's my template strand on top. All right, it's this one here. And we are going to read the DNA 3 to 5 in this direction. And we will make my new RNA 5 to 3. Note, this is a mistake here, right? This should be the 3' end of this one, okay? Now, transcription requires something called a promoter. And a promoter is simply a sequence on the DNA that is recognized by a protein, which allows RNA transcription to occur. So here's my promoter site, and here's my gene in between, and here is my terminator. All right, here's a promoter. Right, here's my gene. If this is my template strand, and here's my terminator. All right, so the promoter is simply basically a start site, and it has a specific sequence of DNA that is recognized by the enzyme called RNA polymerase, which is what is going to synthesize our RNA. So, what's going to happen then is, if we look at this, here's my promoter. We can have enhancer sequences that are upstream from my promoter, and they allow transcriptional activator proteins to bind. General transcription factors will also bind at my promoter. Right, so we have two things going on. We have enhancers binding at enhancer sequences. We have transcriptional activator proteins binding to those enhancers. And we have general transcription factors binding at the transcriptional start site. Okay? This then allows for RNA polymerase to come in, right here. This is a mediator complex. And it allows the RNA polymerase to properly engage right here at my promoter. All right? So here you can see the enhancer sequences, right? And those activator proteins binding to the mediator complex. Right? Here are my transcription factors, and here is my RNA polymerase 2. So all of this is binding. And then what is going to happen is that my RNA polymerase is going to use the template strand up here at the top, because I'm moving from left to right, so I'm heading in this direction. So this one is 3 to 5, so we read the DNA 3 to 5. Here's my 5' end of my RNA. Here's my 3' end of my RNA. So this enzyme is just going to work its way this direction. It's going to unzip the DNA. It will synthesize the RNA right here. It's complementary. And then it has basically a keel or a wedge that will separate the RNA from the DNA that we're making. All right. So let's have a look. Here's my template strand. So if we're going 3 to 5, we're synthesizing this thing in this direction. All right. We are reading the DNA going this way, and we are adding the new base here. All right, the new nucleotide right here. So notice, here's my 5', here's my 3' hydroxyl. We're going to form that phosphodiester bond. We cleave the two phosphates off of it, all right, and we end up with a phosphodiester bond. Each of those shown here. All right, that's a phosphodiester bond. How do we know which nucleotide to put in here? It's determined by the base, right? Which base is the complement to the DNA base. All right, so one more time real quick. 3 to 5, we're reading the DNA. 5 to 3, we're synthesizing the RNA. Here we are adding onto the 3' end from our 5' phosphate. How do we know which nucleotide to put in? It's determined by the base's complementary base pairing. All right, so if we look at this then, here's my DNA, all right, and we're making it single-stranded. So here's my template strand, here's my RNA transcript coming out, And here are the trinucleotides. So we want to think of the trinucleotides as A's, U's, G's, and C's. So how does it know that, say right here, that we are putting a G in? How do we know to place a G right in that location? We know that because at that location on the DNA is a C. So we follow the complementary base pairing rules, and that dictates which nucleotide is placed in series. Alright, so that should wrap up the basics of transcription. I know we went quickly. Bring your questions to class.